Welcome to our unit on cell growth and division. So let's talk first about why cells need to divide. First of all, most cells in your body do divide regularly, and there are a couple of reasons for that. If cells didn't divide, there'd be two major problems. The instructions in the DNA would be overloaded trying to produce proteins the cell needed. So as we've learned, DNA produces proteins, and so in order to make all the proteins necessary for a very large cell, we would need a lot of DNA, or the DNA that was available in the cell would need to be copied constantly. And so there's just not enough of that template. Think of it as a blueprint. If you had one construction manager with all the blueprints for all the construction crews in the United States, that would be an overload on that one set of blueprints. So same thing occurs with DNA. We need to have fewer instructions necessary, so fewer proteins made. So we're going to keep the cells small for the DNA to be used efficiently. Secondly, the cell would not be able to exchange materials quickly enough for its needs. So let's look a little more closely at that. If you picture this as a cell here, obviously it's a cube, but if this is the nucleus and this is the membrane, the cell membrane outside of the cell, look at the difference here between the nucleus and the cell membrane as opposed to this smaller cell that has a nucleus and a much smaller cell membrane. In order to move materials in and out of that cell membrane efficiently from the nucleus or to the nucleus or any other part of the cell for that matter, it's going to be a lot more efficient to move things in a small cell like this. So in order to solve the problems that I just mentioned, the cells keep a large surface area to volume ratio. So this cell here has a low surface area to volume ratio. This cell, the smaller cell, has a high surface area to volume ratio. So what does that mean? These are terms that you've probably learned in math class, but just as a review, to calculate surface area, you take the area of one side, one square of this cube, if we had a cube-shaped cell, and you're going to square that to find the area of this one side. So 5 centimeters squared times 6 sides, since we have 6 sides to our cube. And that's going to give you a surface area, total outside area of this cell membrane of 150 centimeters. Sorry, that should be square centimeters. The volume is going to be length times width times height for a cube. Those are all the same. And so our volume is 125 cubic centimeters. So if we divide that out, surface area to volume, we're going to get a 1.2 to 1 ratio. So 150 divided by 125 is a 1.2 to 1 ratio. So in other words, the surface area of the cell and the volume are very similar. For this smaller cell, if it's only got a 1 centimeter side, if we have a cube-shaped cell, the surface area is going to be 6 square centimeters, and the volume is going to be 1 cubic centimeter. So that's going to divide out to give us a ratio of 6 to 1. So this is a much higher or larger surface area to volume ratio, which means that in order to efficiently move materials into this cellular area, into the cytoplasm area where all the organelles are located, this cell is going to be able to do that very efficiently because there's a really large surface area here that it can move things in and out. I know this seems like it has a larger surface area, which it obviously does, but it's servicing a huge interior of the cell. And so it's not going to have as much space to move molecules across because, as we learned, the cell membrane is responsible for moving materials across, and sometimes that requires proteins. So there needs to be enough proteins to service this entire intersection of the cell. So the high surface area to volume ratio works a lot better for moving materials efficiently. So how do organisms reproduce? Cellular division is one of the ways that organisms reproduce. And as we've established with our characteristics of life, all living things must reproduce somehow. Most unicellular organisms reproduce through asexual reproduction, in which one cell divides into two. That's what we'll be talking about most in this unit. So asexual means that there's only one organism that is used as a parent. It does not require two parents. Multicellular organisms can also reproduce asexually, which isn't something that you think about regularly. But they can asexually reproduce through budding, like this hydra, which is an aquatic animal and similar to a jellyfish, if you think of it as an upside-down jellyfish, sort of. This bud can actually form asexually off of the hydra, the adult hydra, and then it grows out into a separate organism, eventually breaks off and leaves. Vegetative propagation is something you might be familiar with. Some portions of certain plants can be cut off and regrown into a whole new plant. Sometimes you might do this with vegetables in your garden or in your house. You can actually cut off pieces of vegetables and have it grow into a new plant. Sometimes we do that with plants that have aerated roots. If there's some aerial roots that are hanging off of your house plant, you can break that off and replant those and they'll grow into a separate plant. That's vegetative propagation. So both of those are ways that multicellular organisms can reproduce by themselves, asexually. Sexual reproduction, as you know, involves the joining of two reproductive cells, one from each of two parents. So here we've got a sperm cell and an egg cell, and as those join, these would be, let's say, human sperm and eggs. They are going to join genetic information. So although asexual reproduction is often easier, of course, because there's no need to find somebody to reproduce with, sexual reproduction helps to mix genetic information, which can improve a species' survival. So asexual reproduction creates an identical offspring that's identical to the parent, so the genes are going to remain the same. But in sexual reproduction, these two parents' genes are actually going to combine, and we'll talk more about that in future units, how that works, but they combine to mix up the genetic information. So each offspring is then going to be distinct with a new set of genetics. And that allows, in a population, different individuals to survive and not be wiped out by all of the same environmental conditions or disease, because they are all slightly different. Okay, so let's talk about this genetic information. The genetic material in a eukaryotic cell is found in the nucleus. We learned that back when we talked about the different structures in a cell. During most of the cell's life, it is arranged in long strands called chromatin. So here in our very simplified picture would be these blue strands. When a cell divides, the chromatin is wrapped up to prevent it from getting tangled and broken. Because when a cell pulls apart, you don't want those long strands to get mixed up or wrapped up or somehow broken and wrecked, because the DNA is so important. So in order to protect it, the DNA is wrapped around proteins called histones and then organized into X-shaped chromosomes. So here's the long strands of DNA. These are the histones that it winds around, and then it condenses around those histones and forms this X-shaped chromosome. Prokaryotic cells have one circular piece of DNA that does not organize into chromosomes. So this is part of the less complex structure of a prokaryotic cell. It doesn't have the histones, it doesn't form chromosomes, it just keeps those long strands of DNA, and it's really just one big circular piece. So in summary, cells need to divide to use DNA and pass materials more efficiently. Organisms can reproduce through asexual or sexual reproduction. When cells divide, they organize their DNA into tightly packed chromosomes. So now that we've discussed when cells divide and why they need to, we'll discuss the process of cell division, what is actually involved. So starting with prokaryotic cells, which remember are the cells that don't include membrane-bound organelles, like bacteria. Prokaryotic cells divide by making a copy of their DNA and then pinching the cell in half. So you can see that happening here. This is the circular DNA. It copies itself, doubling, and then the cell pinches in half. That process is known as binary fission, and it results in two identical cells because the DNA is copied. In the eukaryotic cell, the process is a little bit more complicated. Most of the cell cycle for a eukaryotic cell is spent in interphase, when the cell is growing. So interphase is represented by this orange section here on our cell cycle diagram. And there is this little outgrowth here called G0, and that would be when the cell is resting and it's not preparing at all for division. The rest of this time in interphase, G1, S, and G2, out of this orange section, are all preparation for division. And only a small percentage of the time is spent in mitosis, which is this small section here. This is when the cell would actually be dividing. The rest of this time, it's preparing for division, unless it's in G0 and resting. So interphase is divided into these three parts. G1, in which the cell grows in volume and it duplicates its organelles, because not only does the DNA need to be replicated and prepared for division, but also all the organelles need to be replicated, because if there's only one set of organelles, then when the cell divides, there's not going to be enough for each of the two new cells. In S phase, the cell copies its chromosomes. And so that's where the DNA portion is copied. And then in G2, the cell continues to grow and it checks the duplicated chromosomes. So it's making sure that there weren't any mutations that occurred while that DNA was being copied. Why is it important for the chromosomes to be copied? As I mentioned, they must be copied so each new cell has the same genetic information as the original cell. So in this process of DNA replication here, these are our chromosomes. They double and form those X-shaped structures that we talked about before. And then through the process of mitosis, they're going to split so that each of the two new cells receives a copy. So what are the phases of mitosis? Remember, this is a very small portion of the cell's life, but during this time there are several phases. The four main phases involve splitting the cell's chromosomes, because that is the most important part for the cell, to make sure that the chromosomes are duplicated and split correctly, because that's where all of the instructions for the new cells will come from. And the four main phases are prophase, metaphase, anaphase, and telophase. And then additionally, after the chromosomes are split, there's a process called cytokinesis, which is the splitting of the cytoplasm, the rest of the cell. You can see a brief overview here of those different phases at the bottom. Okay, going into more detail on each of these phases of mitosis, we'll start with prophase. In prophase, the chromatin, which is those long strands of DNA, begins to coil into chromosomes. And those chromosomes are X-shaped structures because they're duplicated DNA. They've already duplicated during interphase, and each of these sides of the chromosome is called a sister chromatid, and they are identical copies of each other. Also during this process, the nuclear envelope begins to disappear. So remember, there's a membrane around the nucleus, and that begins to start to degrade, and that's going to release those chromosomes so they can move around the cell more freely. Also, there are centrioles that move to the poles, and the centrioles and the spindle fibers are part of the cytoskeleton of the cell. So the centrioles begin to move to the poles or the ends of the cell, and the spindle fibers, which are these threads, begin to form between them, and they will help to move the chromosomes into the correct location. In metaphase, chromosomes attach by centromeres to the spindle fibers. So again, here's these spindle fibers responsible for locating the chromosomes and lining them up correctly in the cell. And those chromosomes are going to be lined up in the middle, which is a good way to remember metaphase, middle. And as I mentioned, each duplicated chromosome here is made of two identical sister chromatids attached at a centromere. So centromere is the middle of the chromosome, and then each of these sides is a sister chromatid made of a copy of DNA. In anaphase, the next phase of mitosis, the centromeres of the chromosomes split, and the sister chromatids are pulled to the opposite ends of the cell. So they're being pulled away from each other. These copies of the DNA are being separated so that each new cell will receive a copy. In telophase, chromatids reach the poles, or the ends of the cell, and the nuclear envelope begins to form around them. Because remember, we need to keep the DNA protected whenever possible. The spindle fibers start to disappear, and the cell is going to begin to elongate here, finishing with cytokinesis, which divides the cytoplasm. So now the DNA has divided, and then the rest of the cell, the cytoplasm and all the organelles, begin to divide during cytokinesis. This is different in plant and animal cells. In animal cells, the cell membrane pinches together, just like you saw in binary fission. But in plant cells, because they have a cell wall, it's a different process, and what forms here is a cell plate. It begins to divide the cell, and as the cell elongates, that cell plate is going to form in the middle until there are finally two new plant cells. The result of mitosis is two identical daughter cells which are identical to the parent cell. So these two daughter cells are identical to each other, and they're also identical to the parent cell where the whole process began. So the process of mitosis, the goal is to make identical cells. So in summary, prokaryotic cells divide through binary fission, but eukaryotic cells divide through mitosis. Mitosis has 4 phases that result in 2 identical daughter cells. So why does the cell cycle need to be controlled? We've talked about the phases of the cell cycle. Now let's talk about how it's regulated. Cells grow and divide at different rates. For example, a fly embryo cells divide every 8 minutes, which is incredibly rapid. On the other hand, mammalian liver cells divide only once every 1 or over 1 year. So this is a wide spectrum of speeds at which the cell cycle can move. And the cell cycle is controlled by internal and external proteins that allow growth and healing without overpopulating their space or exhausting their available nutrients. If you look at the bottom of this slide here, you'll see some cells growing in culture in a dish. And scientists have determined that cells will naturally divide until the bottom of a culture dish is covered in one layer of cells. And at that point, they stop growing. Rather than piling up and overflowing the dish, they stop growing at that point. Which is good, because can you imagine if your body was trying to heal a cut in your skin and the cells just kept dividing and creating more skin? You would end up with a very different looking skin than you started with. So this is good, but we need to go through the different ways that the cell cycle can be controlled, and then we'll talk about some ways that it can go wrong. So the cell cycle is regulated from the outside. We said it's regulated externally and internally. External regulatory proteins control the cell cycle by receiving signals on the cell membrane. And you can see in this picture here, there are receptors on the cell membrane, and those receptors receive signals from neighboring cells that determine whether the cell should continue growing and dividing or whether it should stop. So these signals can speed up the cell cycle, and those are called growth factors, or they can slow down the cell cycle when an area is too crowded. On the other hand, on the inside of the cell cycle is regulated by a group of proteins called cyclins, and they regulate the timing of the cell cycle from the inside. So at several checkpoints along the cell cycle, these cyclin proteins join with a protein complex known as CDK, or cyclin-dependent kinases. And if the cell clears that checkpoint, the cyclins degrade until they're needed again. So let's see what that looks like. This is a complicated image here, but let's go through what's happening. We have the CDK molecules in this cell cycle, right? So the cell is growing here in G1, and it is duplicating its organelles. And that CDK joins with cyclins. There are multiple cyclins in the cell process, and those cyclins join with the CDK complex, and if that continues, then if that checkpoint is passed, they're able to actually break down proteins and other substances that are blocking the cell cycle. So this is a good progress point for the cell cycle. And then after that checkpoint is passed, the cyclins degrade, and CDK can be used with a different cyclin. And so there is one checkpoint here at the end of G1 where DNA damage is checked for. And if there is DNA damage, then the cell cycle will stop at that checkpoint. There's another checkpoint in the S phase, at the end of the S phase, and at that point, if the cell has not correctly replicated its DNA during this S phase like it should have, then the cell cycle will stop. And then there's another checkpoint in mitosis that allows the cell cycle to continue. And this third checkpoint is a spindle assembly checkpoint. As you know, spindles are necessary for moving chromosomes around the cell during mitosis. So if the spindles are not correctly assembled, then that will stop the cell cycle as well. So if the cell fails a checkpoint, an inhibitor molecule will attach to the CDK cyclin complex, and it will stop the cell cycle. So this complex is needed to keep the cell cycle moving. And if there's a problem with one of those checkpoints, the DNA hasn't been correctly replicated or the spindles aren't correctly formed, an inhibitor molecule will join that complex and the cell cycle will stop. So in summary, the cell cycle is regulated externally by signals from other cells and internally by the formation and degradation of cyclins that occur at certain checkpoints along the cycle. So what happens when the cell cycle is disrupted? As we just established, normally the cell cycle stops when the cell runs out of space or runs out of nutrients and it needs to stop dividing. But what happens when that doesn't work correctly? Cells that fail to stop at the required checkpoints can divide too rapidly and live much longer than normal cells. That makes sense because this process is supposed to regulate how quickly the cell is growing and dividing. So if they're not stopping correctly at those checkpoints, then the cell is going to divide rapidly and live longer than it normally would. And while some tumors are harmless, others are malignant or cancer-causing. And malignant tumors can spread to other parts of the body in a process called metastasis. So if we have a tumor here and the tumor cells are located just outside some sort of blood vessel, which would be pretty typical because you have blood vessels all over your body. If a tumor cell breaks off and begins to enter the bloodstream, it can travel to a different part of the body and it can reintegrate in a different tissue, giving rise to a new tumor somewhere else in the body. So that is how tumors can spread. What causes cancer exactly? So when pieces of DNA are damaged, they do not correctly code for the proteins that regulate the cell cycle. So there's, as we've established, there are many proteins involved in regulating that cell cycle. And when DNA is damaged, then they don't code correctly for those proteins, or they become abnormally shaped proteins that don't work correctly, and they don't regulate the cell cycle the way they're supposed to. This DNA damage can be caused by exposure to carcinogens like tobacco, radiation, or human-made chemicals, and even viruses. So here's an example of of damage that's been done through skin cancer. Melanoma is a type of skin cancer, and it's caused by exposure to UV radiation. So UV radiation from the sun has hit these skin cells and damaged the DNA, and now that DNA does not code properly for the proteins that regulate the cell cycle, and that cell cycle continues abnormally. How is cancer treated? So tumors can be treated in a few ways. Surgery. If the tumor is localized and hasn't spread, it can be surgically removed. And radiation and chemotherapy. These are treatments that kill rapidly dividing cells through targeted beams of radiation or specialized chemicals. And there are immune therapies and hormonal therapies that are also used for cancer that's spread. And radiation can also be used for a localized tumor, but surgery is one of the best options to remove a tumor if it hasn't spread to the rest of the body. So in summary, if the cell does not correctly stop at the checkpoints in the cell cycle, it can become cancerous or malignant. These abnormal cells can form tumors, which may metastasize and spread to other parts of the body. So we've discussed how cells grow and divide. Let's talk about how we get the different types of cells that are in your body. So you have about 200 different types of specialized cells in your body, For example, stomach cells, muscle cells, and nerve cells. And all of those different types of cells have different characteristics and different functions. But they all originate from stem cells. A stem cell is an unspecialized cell that can divide indefinitely and become many different types of cells. So let's look at how that process happens. The process of forming specialized cells is called differentiation. And multicellular organisms like plants and animals begin as one cell that divides into a small clump of cells called an embryo. The cells within an embryo are totipotent, meaning they can differentiate into any type of cell in the body. So here's the fertilization process, sperm and egg coming together, and an embryo is formed from that as the cells begin to divide. And as the organism continues to develop, the cells specialize more and more, and they have a more limited potential to differentiate. So these embryonic cells will become all the different types of tissues in the body, and they lose their potential to become different types of cells as they start to specialize. So they start as totipotent cells in embryos, and then the embryo has a couple of different layers in it, and each of those layers becomes pluripotent, meaning it can become only a few different types of tissues, and then those cells become multipotent, eventually leading to specialized cells which only have one function, or unipotent. So how can stem cells be used to our advantage? Because of their ability to differentiate into other types of cells, stem cells can be used to replace damaged tissues or repair organs that have been destroyed by disease. And this is known as stem cell therapy or regenerative medicine. So here's the way this works. If stem cells are collected from bone marrow, because adults do have some remaining stem cells, and stem cells can be collected then from the bones, and they are then reprogrammed to grow into different types of tissues and then implanted back into the patient that has a disease or has lost the use of organs for a certain reason. So what role does DNA play in this differentiation process? Well, first of all body cells contain the same DNA. So even though you have different types of specialized cells in your body, they all have the same DNA. Each specialized cell only uses sections of DNA that create proteins it needs to function. So it's only using a portion of that DNA that provides for its structure and function. So each band on this chromosome represents a section of DNA that codes for a protein. Different types of cells use different sections from the DNA. Muscle cells might use some of the sections, while nerve cells might use others. In summary, embryonic stem cells can differentiate into all types of specialized cells in the body, using different sections of DNA for each type. Stem cells can also regenerate tissues that have been damaged.